After data loading, the next step normally we do is called QC, quality control. The quality control primarily involves two steps. One is to filter cells with incredibly high amount of mitochondria gene expression. The second one would be to filter out cells with either incredibly high amount of RNA number or very low number of RNA expression. For individual cells, it would have both mitochondria gene and nuclear genes. A high amount of mitochondria gene would indicate the cell stress or apoptosis marker, compromised cellular integrity, or technique artifact. The high amount of mitochondria gene indicate the cell is in a bad condition, so we want to filter those cells out of the data. Normally we use 5% as a threshold to filter out the cells. For human data, the mitochondria gene normally start with capital MT. The mouse data normally start with lowercase mt, stands for mitochondria. We would also filter out cells with incredibly high amount of RNAs or very low amount of RNA. This is also standard to filter out the abnormal expressed cells. To do this, let's go to the scRNAt. In our previous video, we have talked about how to load the scRNAt as well as load the data into the environment. Now we're going to do the QC matrix. Use the column names, scRNAt object at metadata. We can check the number of count RNAs and number of feature RNAs for each individual cell. To calculate the percentage of mitochondria genes for each individual cell, because the data comes from the human peripheral blood sample, the mitochondria gene would start with a capital MT. And we can use a head MT to indicate any empty gene that start with like MT. To find all the genes in this data set, we can say this is a pattern equal MT. And the pattern has to be found in the data set. The data is scRNAt object. We can use a specific function called percentage feature set. This is the function we can calculate MT related genes and its percentage. And let's click run. We'll see it calculate the percentage of mitochondria for each individual cell. We can use head function to see the first few samples. For the first 6 cells, this is the percentage of mitochondria, which is lower than 5, indicating it's in good condition. I want to assign this value to the metadata. As we mentioned, we can use head scRNAt object at metadata to show the metadata for the first 5 cells. Now we can assign this calculated mitochondria value, scRNAt object. I want to assign this value as a metadata and as a new column. It's named as percentage mitochondria, gonna equal to the calculated mitochondria percentage. Once we finish this one, we can check again first 5 samples and its metadata. We'll see the percentage of mitochondria can be calculated for all the samples. Next, we'll do the filtering. We want to subset the samples. Mitochondria gene expression level less than 5%. We'll use scRNAt object, gonna assign to the new value with subset. The data we'll subset is called scRNAt object. And the standard we'll use is called a subset rule. The subset rule will be equal to percentage mitochondria less than 5%. So we use this one. We'll see the scRNAt object will change a little bit. So we can check again the scRNAt object. And if you remember, we can do the assay. RNA assay. And you can check. Now we have in total 2,643 cells. And original data has 2,700 cells, which means we filtered out 57 cells that with mitochondrial gene expression higher than 5%. All right, we also need to filter out cells with either incredibly high amount of RNA expression or incredibly low amount of RNA expression. We can use the same function called a scRNAt object. I'm going to use subset function. And the subset function input is also going to be scRNAt object. But this time, the subset rule going to change a little bit. The subset rule going to be number of future RNA needs to be more than 200. We can further define in a range. So it could be more than 200. At the same time, the number of future RNA less than 2500 picogram. Then we can check the surrogate object RNA, the assay again. It will show the basic description. This time we filtered out additional couple of cells. So now we have 2638 cells left. All right, so this is the basic quality control we're gonna do.